Hello and welcome to the Synthesis Project. I'm your host, Elaine. I undertook this assignment because I wanted to see if my hypothesis of the different models would be correct. I hope to learn more about the environments of microcosms. I believe microcosms can be used in ecology by predicting the behavior of ecosystems under controlled conditions. This presentation will be on 8 separate models involving environmental fluctuations as well as changes in biodiversities. My first model represents the Endler's groupies of koi fish. This model gives you the tools to manipulate population selection, mutation, migration, and non-random mating that violates the assumptions of Hardy-Weinberg's theory. I use this model to find the outcome of koi population when modifying the environment. The first parameter that I manipulated was the migration by increasing it by 0.8. The major finding that I saw was that capital RR fish, also known as mutant fish, increased while lowercase rr and uppercase r lowercase r fish decreased. The second parameter that I manipulated was the number of mutant fish by increasing it, also known as capital RR fish. The major finding was that almost all koi fish, lowercase rr, uppercase r, lowercase r, and including the mutant fish, capital RR fish, have decreased significantly. The third manipulation I did is called the evolutionary parameter, where I manipulated the migration by setting it at 0.9 and the migrant capital R allele prop at 0.2. I also manipulated the population dynamics that include initial size at 25, carrying capacity at 25, sex ratio female to male at 0.5, mortality rate at 5, blood size at 5, and the initial capital R allele prop at 0.7. My major findings include the capital RR fish disappearing completely, while koi fish, capital R lowercase r, and lowercase rr koi fish stayed present. The most dominating fish was the lowercase rr koi fish, also seen as the whitefish. A pro for the koi fish model would be that it was easy to navigate and change the different populations, though a con would be that I was unable to change the environment itself. An ecological implication for the model of koi fish involved the dump of 3,000 koi fish into a Colorado lake. These fish then started to wipe out the native species and altered their environment. If you would like to know more about the koi fish, there will be links at the end of my presentation with all of the different models that I talk about. My second model is called the random genetic effects. This model provides an illustration of random genetic drifts, bottleneck effects, and founder effects. There are 3 dominating populations: red, yellow, and blue. You are able to adjust the mainland population size and also put them through a bottleneck. You are also able to colonize the 2 islands off of the coast of the mainland with any random subject of the mainland population. The first parameter that I manipulated was by increasing the founding -n-1 to 23 and increasing the founding -n-2 to 50, with the IL-2-K at 200. One major finding was that the color yellow disappeared completely, while the colors red, purple, and blue stayed present. Red was the most dominating color at 0.854, and the least dominating color became blue at 0.146. The second parameter that I manipulated was the increase of just colony 1 IL-1-K to 200. The major finding that I saw was a new color that looked like orange along with the purple present. The red color was the most dominating with a number of 0.727. Blue is the second dominant with the number 0.199 and yellow at 0.074. For the third parameter, I increased only colony 2, founding at 50 in the IL-2-K at 200. Some major findings include the blue color being most dominant at 0.68 with the purple, and the red color being least dominant with a 0.32, while the yellow is completely wiped out once again. An ecological implication for the model random genetic effects can be shown through the extinction of species as well as the recolonization with a population of local species and migrants. For my third model, I have chosen the vigilance behavior model collective. This model focuses on predator and prey. A hawk comes to eat the birds while the birds find food. You are able to change the foraging rate and adjust the duration of foraging. The model also shows the capacity of an individual bird's behavior. For my first manipulation, I increased the food parameters at 0.16 with 16 juncos, a detect at 0.26, spacing at 10, prop forage at 0.44, and a forage time at 5. Some major findings included that 2 birds were killed and 1 was missed. I also noticed that the birds hardly ran away once seeing the hawk. Instead, they kept foraging for food. For my second parameter, I changed the food density to 0.3, the detection to 0.26, and the spacing at 0. My major findings include the birds becoming hyper alert to their surroundings. Less birds were killed because they were spread out from each other. For my third parameter, I manipulated the food density at 15, detection at 0.5, warning at 0, forage time at 25, and prop forage at 1. One major finding was that the birds once again had no detection of the hawk above them, though once every 10 times that the hawk came, the birds would fly away. A pro for the vigilance collection would be that the controls were simple to use. Some cons would be that the model itself was rather boring, and I found some glitches when loading the model. An ecological implication for the model of vigilance in birds can be shown in an article that talks about the potential risks that birds have when going to a feeder. This article looks at the waiting time and the foraging time that the birds give themselves in order to not be caught by their predators. For my fourth model, I have chosen the Endler's groupies. This model focuses on a 1980s classic experiment on the balance of sexual selection and the natural selection. The females usually prefer males with a lot of dots, though males with more dots can easily be spotted by predators. In this model, you can manipulate the strength of the female preference and the number of predators. For my first manipulation, I increased the number of mutant fish to 0.0041, an increase with the fish to 69 for a groupie, and Gunny K at 18. The running time was also set to 20,000. Some major findings that I saw was that the distribution of spots increased while the female percentage decreased. For my second manipulation, I increased the number of Gunny K to 228, morality at 0.4, mutation at 0, and the blood size at 10. I kept the running time at 20,000. My major findings included a huge increase in distribution of spots, a decrease in average number of spots, and an increase of female population from 39.1% to a whopping 60.7%. For my third change, I chose to lower the Gunny K to 162 and add 7 num Rivius. Some major findings include a decrease in distribution of spots, a steady average of 6 spots, and the percentage of female decreasing. A pro for the Endler's Goopy model include having many controls to manipulate so that I could see many outcomes. Though a con would be the model itself was very busy and it made it kind of confusing to understand. Ecological implication for the model Endler's Goopy would be the altercation of mortality rate as well as direct and indirect effects of the species throughout its history of evolution. This specific article looks into this species called Revelis harti. For my fifth model, I have chosen the honeybee foraging. This model focuses on the stimulation of foraging behavior of honeybees. It is made to illustrate Charnova's 1976 marginals valve theorem. The model gives you the opportunity to change the forage time and modify the environment's condition. You are able to change how far or near you want the flowers. The model collects the total nectar taken and gives a rate of the nectar collected. For my first manipulation, I increased the flower distance to 26, B1 forage time to 140, while B2 forage time to 65. I also changed the nectar AMT to 10 and the extraction rate to 0.02. Some major findings included B2 being much faster than B1, though the collection rate for both bees stayed the same as one another throughout the collection. For my second manipulation, I decided to decrease B1's forage time to 50 and B2 forage time to 150. The major findings showed that B1 was incredibly faster compared to B2. However, the collection rate was slightly different with B1 at 0.0205 and B2 at 0.0235. For my third manipulation, I changed the flower distance to 6 with B1 forage time at 110 and B2 forage time at 150. I also changed the nectar AMT to 40. One major finding was that the collection rate went up significantly due to the bees being much closer to the flowers. Another finding showed that B1 was much faster than B2 with a collection rate at 0.0530 and B2 having a collection rate at 0.0457. A pro for the honeybee foraging would be that the model was very easy to understand and therefore was easy to use, though a con would be that it was super boring and I did not have many controls to manipulate. I also felt that the outcome was easy to predict. An Ecological implication of the model honeybee foraging can be shown in an article called Proceedings of the Royal Society Bee that focuses on the different structures of landscape and how it can affect the bees foraging. For my sixth model, I have chosen the barnacle competition. The model is a Connell's 1961 classical competition experiment. You are able to observe two different species of the barnacles called the Cathomalus and the Balamanus. One of the species is able to grow at a much wider range in depth, and the other is limited to deeper rocks. You are able to manipulate the tide as well as the sea levels to show change in global warming. For my first manipulation, I changed the sea levels to increase at 3 meters. The tide change was also increased at 6 meters. The Balamanus and the Cathomalus stayed the same at the value of 2.5 milliliters, as well as the Thais density, which is at 3. A major find I saw was that the Balamanus increased in population and in size on top of the stump. However, the Cathomalus was unable to grow big and therefore decreased in numbers and in size. Most barnacles continue to grow above the low tide because the predators were unable to reach them. My second manipulation involved the changing of Cathymus with a value of 5 and the Balamus at a value of 1. I decreased the sea level to 0.5, the tide changed to 1 meter, and the Thais at the value of 3. A major finding was how the Balamus acted as a border between the Cathymus and the predator. Because of this, the Cathymus was able to grow in numbers but not grow big. The Balamus was able to grow big but not in numbers. Instead, the Balamus decreased in numbers. For my third experiment, I changed the tide to 6, the Balamus to 5, the Cathymus to 5, and the Thais to 8. Some major findings showed the top half full of Cathomalus and the bottom half full of Balamanus. Both barnacles increased in population. The Cathomalus stayed small while the Balamanus grew big. Because the tide change went very low, the predators were unable to reach the barnacles, and therefore both barnacles were able to increase in numbers. A pro that I found for the barnacle competition model would be that there were just enough controls to make it easy to change the environment. I also found the model aesthetically pleasing to the eyes. It was very fun to use and fun to see the outcomes, though some of the outcomes were very easy to predict. An ecological implication of the model barnacle competition can be shown in real life from an article called The Competition Between Barnacles. The article focuses on the Cathomalus and the Balamanois found on the rocky shores of Scotland. For my seventh model, I decided to focus on microchromosomes. This model is made from Gauss's 1934 classical experiment with process. The model is a virtual petri dish that allows you to add 2 species of Paramecium and a predator. One of the species is a much better competitor for bacteria. The other species has photosynthetic endosymbionts that utilizes light, though both species is a prey for the Didinium. This model is used to observe how the 2 species react to one another and their predators. The first experiment I conducted was by adding init bacteria of 200 and adding P. aurelia into the mix. It wasn't long before the bacteria increased in numbers as well as the P. aurelia. The bacteria count got to 1,774, and the P. aurelia increased by 74 in just 947 ticks. The count of P. aurelia started at 50, and the bacteria started at 201. For my second experiment, I increased light to 9 and decreased volume to 4. I also added 35 Berseria with 5 didymium. To my surprise, the bacteria and didymium both disappeared entirely, though I saw that the first to disappear was the P. aurelia and then the didymium. The last manipulation involved decreasing the light to 4 and increasing the volume to 10. I added 201 bacterias and 80 Berserias. My major findings were that the bacteria steadily increased while the Berseria decreased. After just 344 ticks, the bacteria was up to 2,117 and the Berseria was down to 46. A pro for the microcosm model would be that they gave many different controls, and I was able to add as many species or predators that I wanted to the mix. A con would be that the model was very busy as well, so it was kind of confusing to figure out which ones were bacterias and which others were the species of parasemiums. An ecological implication of the model microcosms can be found in real life through an article called Microcosms Experiments Can Inform Global Ecological Problems. The article goes into depth about how a small-scale experiment using a model organism to help prevent or see an outcome of a climate change or manage biodiversity. For my final model, I have chosen the random genetic drift. The model focuses on the adaptation of a classic experiment made by Peter Bury in 1956. This experiment documents the genetic drift in laboratory population of Drosophila. This model holds 10 valves of flies with a constant population size and has proportions of a mutant allele that are tracked through generations. The population size and the initial allele proportions can be modified. This experiment is used to document the fly adaptation in genetic drifts. For the first manipulation, I decided to change the population size to 10 and the initial prop/BW to 0.9. A major finding was that the generation increased to 85, colonies fixed became 10, the mean fixed time became 84, and the overall population-BW stayed at 0.9. For my second experiment, I set the population size to 80 and the overall prop-BW at 0.25. The major finds I saw included a particular spike in pen-7 at 0.725, then decreased at 0.28. I also saw that the over-prop-BW became 0.11, generation became 117, colonies became 6, and the mean fixed time was at 35.6. For my final manipulation, I changed population size to 20 and the initial prop-BW at 0.1. A major finding was that the overall prop-BW became 0. The mean fixed time was 20.2, while generation was only 72, though all 10 colonies were fixed. A pro that I liked for the model random genetic drift was the fact that I could see a chart of all the fish valves. Though a con would be that I also found the chart very confusing and all over the place when running my experiment. Therefore, I tried to do a more simple test. An ecological implication of the model random genetic drifts can be shown in an article called The Genetic Drift. This article goes into depth about how the drift of genetics all depends on the population size. In conclusion, I have many shocking moments with different outcomes of the model experiments and manipulations. For example, in the second outcome of the honeybee forager, I thought bee 2 would collect more nectar due to it being slow, though it showed that the first bee was much quicker and better at collecting the nectar. Another example is with the barnacles in the third parameter manipulation. I wanted to create larger cathomeluses, though the cathomeluses would not grow bigger than the balanus. I also noticed that the balanus only grew under the sea level, while the cathomelus mostly grew above sea level. Overall, I was very pleased with my findings, and I hope you enjoyed this presentation. Here are all my references.